on your presentation. Well, now moving on with our presentations, I'd like to invite as the chair of the next panel, Advanced Therapy and Public Health and Unified System Technology and Access, the coordinator of the National Institute of Science and Technology and Medicine, Regenerative Medicine, and the National Network of Cellular Therapy, Antonio Carlos Campos de Carvalho. Is a physician from the Federal University of Rio de Janeiro. He has a PhD and a master's in biological sciences and biophysics from the same institution. He has had ample experience in physiology and biophysics with emphasis on cardiovascular physiology, acting mainly on the following topics: communication, intracellular communication of stem cells and cellular therapies. He was a visiting professor at the Einstein College in New York. He is an emeritus professor at UFRJ, a member of the Brazilian Academy of Sciences and the Academy of Sciences in the Developing World. As the debater, I'll invite the Vice President of Production and Innovation in Health at Fiocruz, Marcus Aurelio Krieger, who will be coming to talk to us virtually. He'll be speaking to us on video. Has a PhD in biophysics from the UFRJ, has a BS from Federal University of Paraná in biology. He has been working in molecular parasitology and biology, molecular biology, working on the trypanosoma cruzi, genetic expression, functional genomics, cellular differentiation, and the use of molecular biology technicians towards the development of tests and diagnoses. It was the adjunct director of development, technological development, prototyping, and production at the Carlos Chagas Institute at Fiocruz. Is a technical coordinator of the production unit at Fiocruz for diagnoses of nucleic acid. As speakers, we would like to invite Professor from Paramount School of Medicine from Pennsylvania University, James Wilson, who will present to us through a video EASI-based vectors as a platform for treating disease. His career has been devoted to rare diseases and ways to treat them through genetic therapy. He's a medical professor and pediatrics and a director of genetics at the University of Pennsylvania. He led efforts to develop the field of genetic therapy. Most recently, he focused on vectors, improving gene therapy and clinical applications, and the addition of genomes and mRNA therapies as well. I would like to invite on stage the co-founder and CEO of the Caring Cross, Boro Dropulic. Please, sir. You'll be making the initial presentation, lentivirus vector-based solutions to treat human diseases.
University of Western.
His PhD from the University of Australia and MBA from Johns Hopkins University. He has had experience in the design and development and the manufacturing and technical translation and regulation, implementation, clinical implementation and commercialization of vector technologies and lens viruses. He founded the company that developed the first genetic therapy product approved by the FDA and co-founded Caring Cross. And researcher from the National Institute of Cancer Institute, Martín Bonamino. Gene therapy-based approach to treat patients at the public health system. That's his presentation. PhD in biological chemistry from UFRJ and BS in biological sciences at the same institution. He has had experience developing genetic therapy protocols and immunotherapy against tumors. In addition to acting on the immunological characterization of cancer, he is a Fiocruz expert, where he is one of the coordinators of the Fio Cancer Network.
Antônio Carlos, por favor.
Antonio Carlos, please, you have the floor. Good afternoon, everyone. I will switch to English because the panel is international. It's
and to be able to introduce leading researchers in the field of advanced therapy products. As all of you know, we're seeing a true revolution in medical care, especially for diseases for which there were no previous effective therapies. And in this panel, we will have two world leaders in the field of gene therapies. One that deals essentially with in vivo gene therapies, and the other one that has developed essentially ex vivo gene therapies. Additionally, we'll have a leader in Brazil in the gene therapy field. As you've seen in the previous presentation by his laboratory, Martín Bonamino is one of the few researchers in Brazil developing gene therapies for the public health system. So not only are they leaders in the field, but they're trying to resolve, I think, the main problem that the field of advanced therapies have right now, which is the price of those therapies. And that's the main reason why Fiocruz has signed already a technology transfer contract with Caring Cross, headed by Professor Dr. Boro Dropulic, and is about to sign such a contract with Dr. Jim Wilson from the University of Pennsylvania. So without further delay, I turn to, I think the presentation, the first presentation will be by Dr. James Wilson from the University of Pennsylvania.
Hello, my name is Jim Wilson, and it's a pleasure to be invited to the 8th International Symposium on Immunobiologicals from Fiocruz. And today I've been asked to talk about AAV gene therapy for orphan diseases. A brief background on AAV vectors. There are 3 generations of AAV vectors. AAV first generation were derived from isolates of AAV discovered in the 1960s from laboratory preparations of adenoviruses. At the turn of the century, my laboratory substantially expanded the repertoire of AAV vectors through the identification of AAV sequences from endogenous primate sources. These vectors have names such as AAV8 and AAV9 and form the basis of the majority of vectors that are currently under clinical development. More recently, there have been efforts directed to the development of AAV vectors based on engineered versions of the AAV 2.0 vectors. Today, I'm going to share with you the data that we've generated in clinical development of AAV 2.0. The talk today focuses on the use of gene therapy to treat CNS disorders. The challenge is delivery. The holy grail of gene therapy for neurologic disorders is injecting a vector into the blood to target the CNS, crossing the blood-brain barrier. That has been a very difficult challenge, so the early aspect of the field evaluated ways to inject vector directly into the brain through multiple injection sites. We've instead focused on broader distribution of AAV by injecting the vector into the cerebral spinal fluid either through a lumbar puncture into the ventricle or into the cisterna magna, and it is the cisterna magna approach that we have used in all of our clinical development programs. When we think about gene therapy, I suggest we should think about it as a platform in which we leverage the power of the delivery and the manufacturing platform across multiple diseases to improve the probability of success, simplify clinical development, decreasing the cost, and improving timelines. Our CNS platform uses a clade F vector HU68, very similar to AV9. The vector is manufactured using an adherent 293-based transfection platform in a iCELLis bioreactor and injected into the cisterna magna to get broad distribution throughout the brain and the spinal cord. The therapeutic applications that we are pursuing are in the treatment of global neuropathic diseases, in which gene correction in a minority of cells of the brain can have an impact on adjacent cells in the so-called bystander effect. We have a number of lysosomal storage diseases in clinical development approaching commercial. A company we started is using this approach for acquired adult dementia that is monogenic. We also have programs ready for the clinic in neurodevelopmental diseases and CNS malignancies. This approach is also useful to target cells of the spinal cord, the dorsal root ganglia, and the motor neurons. And we're developing this for next generation SMA1 and 2/3, Kennedy's disease, and ALS. Our most advanced aspects of the CNS platform are described in this table, and they are diseases that affect storage in the brain, so-called lysosomal storage diseases. All of these are clinical stage. The most advanced is MPS II, that has finished pivotal studies in collaboration with the company REGENXBIO. And we also have phase 1/2 data for MPS I and GM1 that are entering pivotal through REGENXBIO and Passage Bio. And then CREB-A and MLD that have open INDs and are enrolling patients now. I'd like to share with you data from a few of these examples. The first is GM1 gangliosidosis. This is due to a defect in the enzyme beta-galactosidase that leads to accumulation of GM1 as the substrate. This disease has various phenotypes. The most common and most severe are early infantile, diagnosed less than 6 months of age, late infantile, and juvenile. The storage pathology as a result of the accumulation of substrate leads to this kind of picture in the brain that is associated with rapid neurodegeneration. The natural history of infantile GM1 is that developmental milestones are not achieved. There is some somatic manifestations, hepatomegaly and coarse facial features, but as a result of the neurodegeneration, hypotonia, and early death. This is a meta-analysis of many studies evaluating the natural history of infantile GM1. That is a diagnosis less than 12 months of age. And what we have here is onset of the disease early. These are months, but you can see almost complete mortality with 50% mortality somewhere around 20 to 24 months, 2 years of age. So a very significant disease with significant unmet need. We're developing a gene therapy using our platform to show that we can diminish the accumulation of substrate and lead to increased survival. So at the University of Pennsylvania, we conducted a natural history study, a prospective study in early infantile, late infantile, and juvenile, where we evaluated GM1 neurofilament light chain in the CSF and plasma neurofilament light chain. And you can see that the substrate is very highly accumulated in early infantile, less so in late infantile, juvenile. Also a marker of neuro damage, elevated in early infantile, reduced in late infantile and juvenile. And when we look at the survival of early infantile with these accumulated biomarkers, you get complete death by 40 months of age. Late infantile is associated with less mortality, and juvenile even less. So a direct correlation between GM1, neurofilament light chain, and survival, suggesting these would be good biomarkers for an accelerated approval pathway. So I just want to share interim phase 1 data in our clinical trial that we're conducting with Passage Bio, where we have dosed 4 subjects at low dose, 4 at high dose, and now actually this is a medium dose, and then 3 at a higher dose, and measuring the CSF GM1, which is not reduced in low dose, but consistently reduced at the next higher dose, approaching normal levels. When we look at all patients enrolled as with a diagnosis of infantile, less than 12 months of age onset, and compare their survival to natural history, we get complete survival, showing a dramatic clinical impact, and really excited about moving this forward very rapidly into pivotal and then commercial for global access. Another leukodystrophy that we have open studies with, enrolled a subject, is Krabbe, early infantile Krabbe, severe, significant neurodegenerative disease, diagnosis less than 6 months of age, and prognosis death 2 to 5 years of age. This is the enzyme that's deficient in Krabbe, and it leads to an accumulation of substrate, in particular cytosine. As I mentioned, we've dosed the subject so far in enrolling, hope to enroll others soon that showed good biomarker correction. But I'd like to share with you the compelling preclinical data that we generated in support of this clinical trial. And that is a dog model of Krabbe disease that has the same phenotype, early mortality within 2 to 3 months of age, and demyelination, as you can see by this Luxol blue stain, in the brain of the Krabbe dog, but not the wild type, with the absence of staining. Clinically, we've evaluated untreated dogs with Krabbe, normal dogs, and then treated dogs. So in the untreated dogs, that they have reduced nerve conduction velocity, but they need to be euthanized before this progresses. Nerve conduction velocity is normal in the treated and then normal animals. And the untreated animals have an accumulation of CSF cytosine, a great biomarker, whereas this is not accumulated in normal in the AAV treated animals. So what about the phenotypic correction? These are 2 dogs from a litter, one that has Krabbe, as shown in this movie. At 12 months of age, sham treated, you can see the animal's tremulous and trying to move, and lost motor function in its hind legs, and had to be euthanized very soon after we evaluated the animal. A sibling from this litter was treated soon after birth through an ICM, injection of vector, and this is the animal at 82 weeks. You can see no hint at all of motor dysfunction, and essentially a normal, vibrant, happy dog. So it's this kind of stable, efficient correction that supported us moving into the clinic. So I want to talk about a really exciting product that we're developing, and that is an improvement on the drug called Zolgensma, which is an AAV9-based gene therapy to treat SMA type 1. SMA type 1, again, is a very severe early infantile onset disease. It's not a storage disease. It's a disease of a loss of function that affects the motor neurons. Diagnosis before 6 months of age, 75% mortality by 13 months of age, will not achieve milestones. A product was used called AV9, based on a vector discovered in my lab, AV9, correcting the genetic defect in motor neurons by infusing high doses of AV9 into the blood, very high doses. That does lead to a substantial improvement in the prognosis of these patients. So that the dose is very high, 1E14 per kilo. It does help the patients, although associated with really substantial systemic toxicity. And the cost of vector is almost $250,000 per dose, and it's too high to treat the attenuated version called SMA 2 and 3. This is approved in many geographies. The asking price is over $2 million, and some health authorities can negotiate a lower price, but incredibly effective, associated with high cost and a lot of toxicity. So we're in the process of developing a next-gen AAV gene therapy for SMA that is improved in a variety of ways. We have an improved vector, and we are proposing to use an ICM delivery versus an intravenous for SMA1. These are studies done in a non-human primate, injecting a vector intravenously, and then ICM, looking at the central nervous system. ICM delivery with a 10-fold less vector is 50-fold better, and as you would expect, almost 1,000-fold less targeting the liver, indicating a likely better safety profile. We've also optimized the vector so that we've improved the safety of the promoter and put in a sequence that can circumvent another toxicity that's been associated with first-generation products, which is sensory neuron toxicity of the dorsal root ganglia. And we show that our improved vector here shows great survival in comparison to other vectors in an SMA model of mice. So this is an improved efficacy and safety in SMA1. The cost of manufacturing can be reduced tenfold, as the potential to be useful for SMA 2 and 3. We can leverage this experience in other sensory motor neuropathies, SBMA, which is Kennedy's disease, Charcot-Marie-Tooth, and ALS. So to conclude, we're really excited about working with Fiocruz in Brazil and to identify ways to get these products approved and distributed and access to patients. And the way that we hope to do this is to create worldwide centers of excellence for our research and commercial enterprise, where the treatment is performed once at qualified centers, and we partner with governments, key opinion leaders, patient advocacy groups, social media, to try to simplify the sales and marketing and the commercial outlay. And included in our early network are regional centers that we will leverage for a broader access and development, with Philadelphia, United States, Brazil, working with Fiocruz and the Minister of Health, also London, Saudi Arabia, and South Korea. So again, I want to thank you for inviting me to participate in this incredibly exciting symposium, and looking forward to our growing collaboration with key stakeholders in Brazil to bring curative gene therapy to patients suffering from rare diseases that are resident to Brazil. So thank you very much.
Now, thank you, Jim, for an excellent presentation. And now we have Dr. Boro Dropulic presenting as what Caring Cross is doing, and especially what it can do also in collaboration with Fiocruz in Brazil.
Boa tarde. It's a pleasure to be here, and I thank Antonio and the organizers for inviting us, and not only to present here today, but we're so excited at Caring Cross to be able to collaborate with Fiocruz and Bonamino on our collaboration of tech transfer, which I'll tell you about in the presentation. So for those that you don't know, Caring Cross is a non-profit organization, but we really run mostly like a biotech company, in the sense that we develop technologies, implement them, and form collaborations to distribute these technologies to our partners around the world. Okay. Yeah. So this is just to give us a grounding how clinically successful many of these products are. These are the ex vivo cellular gene therapy products, and on the left there you'll see about 6 approvals in the CAR T-cell therapy that have been transformative for many patients that have received them in various oncology indications like leukemia, lymphoma, multiple myeloma. And on the right there you'll see the stem cell gene therapies in which there are treatments for genetic diseases but also for sickle cell and beta thalassemia. We are focused on these autologous therapies because they really work very, very well. What these autologous therapies are, you're taking cells from a patient, you're then genetically modifying them, now usually almost exclusively with a lentiviral vector, and then those cells are then reintroduced back into the patient. And we focused on autologous and single donor therapies. You can have a donor, but in this kind of very fast manufacturing process because these therapies really work well. There are other therapies on the horizon like allogeneic and IPS, but they are very far into the future in terms of how they compare in terms of their safety and efficacy compared to autologous. So we're really focused on autologous and making this paradigm shorter, simple, more economical for delivery to the broadest number of patients that require them. But their current price is very high, right? Kymriah is $475,000, Yescarta $375,000, and Zynteglo, a treatment for beta thalassemia, is $2.8 million. This is an extraordinary high price, and it includes only the product cost, and it's really not sustainable over the long term, even like countries like the US and Europe. Right now, there's a very limited number of patients that are being dosed, but when these therapies become mainstream, tens of thousands of patients, there's not enough money in the system to really support this level of pricing. And it's certainly not feasible in low middle income countries, right, obviously, and it really restricts access, particularly for underserved populations. And those underserved populations are not just in low middle income countries, they're also in the US. And this is just a data point here that was published where we're looking at the CAR T-cell therapy utilization by race in the United States from 2016 and 2021. And what we find is that about 80% of the patients receiving CAR therapy were white, where about the median population in the US that is white is 60%. Clearly disproportionate access against those patients who are underrepresented. And the global distribution beyond the developed nations, the high-income nations, let's say, there's no significant access, as we know, because of the high price. So why is there such a high price? Well, it's a complex logistics. When pharmaceutical companies own these products, they want their own logistical network and systems and their own factories to produce it, and that's quite understandable. And so you're shipping the product. Let me see if I can get this to work. Yes. So this means you're shipping the product from the hospital first to the factory, and then back to the hospital. And usually there's one facility per continent, right? So it's very long logistics. And this complex logistics and the payment logistics that go on between the hospital and the company and the insurers is, with the central manufacturing facility, is very expensive to establish. And because you need such a high number of personnel to maintain these facilities, it's very expensive to maintain, particularly the chain of custody, chain of identity. And because each company wants to make its own infrastructure, its own manufacturing facility, that manufacturing facility has a high capital cost. And that capital cost needs to be put back into the final product cost, and that's why they're very expensive. And the patient result is not all that exceptional, let's say. When you have to cross state and country lines for the approval of these payments, that significantly delays access to patients. Also, vein-to-vein times, it's been reported that in the US, it's 56 to 85 days average. And what that means is that many patients with advanced disease don't get these therapies in time. They die while the therapy is being made. And that's really, I think, unacceptable. And also, it's difficult for patients to travel long distances because of their families to large treatment centers to get access to these products. So right now, it's really not optimal for the patient. So how do we think about reducing cost and enabling affordable access? Well, we think about it in terms of, on the left there, you can see point-of-care manufacturing, where the product is made at the clinical center or very close to the clinical center. Point-of-care manufacturing is significantly much more economical than centralized manufacturing. In fact, it can reduce the cost by a whole magnitude, right? And it certainly lowers the carbon footprint. Also, there are no cross-border payer issues, which means the patients can get the therapy fast. You don't have to go approval between an inter-country payment or an interstate payment, for example, in the US. And the vein-to-vein times are generally very short, about 7 days average, but we can see also how they can be 1 day in the future. And it's better for hospitals because when the hospital is making the product locally, or has access to the product locally, it can schedule the patients better. So those patients that come in with more advanced disease, they can be prioritized compared to those patients with less advanced disease. So it's much better for patient scheduling when you make the product locally. And many will tell you that hospitals can't manufacture these products. It's always companies that manufacture the products. And that's actually not true. Actually, hospitals have had BLA-approved products in the US. In fact, they make cord blood under FDA approval. And they're certainly capable of manufacturing CAR T cells. Many of the centers that pioneered this field manufacture these CAR T cells at their facilities. So it's not just about point of care, but it's also about improving the technology, right? We're focused on short and simple manufacturing processes, in contrast to where many companies are going to very large, sophisticated, automated systems, which actually doesn't help in terms of the cost. The cost's still high. You maybe save a little bit on labor, but a lot of the material cost becomes very, very expensive. So what we're focused on, as you'll see in the presentation, short processes, simple processes to lower the cost, and integrating automation when necessary, right? In some cases, it's really not necessary to produce highly functional, safe products. If it's necessary in the future, we certainly can implement them. So the first thing that, when you think about point of care, that the regulatory authorities will ask is that, can you manufacture products, consistently throughout a network? And the FDA and other regulatory authorities have been looking at this very closely, and they've been very supportive of it. On the left there, you'll see that, in fact, a dossier from the FDA on point of care or distributed manufacturing for drugs, seeing that one site is a very weak supply chain. Having multiple sites is very, very useful for redundancy in the supply chain. And in the middle on the top there, you'll see the sweet spot for point of care manufacturing, and that's autologous cells. When you're taking cells from a patient and putting them back in, well, it's a sweet spot for point of care manufacturing because you want a rapid turnaround time. And there's some other data. If you look on the bottom right there, I'm not going to go into it in great detail, but in Spain, they've already developed a network. They've developed a CD19 CAR that's produced in multiple hospitals around the country and is being insured, paid back. The product is successfully being made. So certainly this is being looked at in other countries and is being successfully implemented. We published a couple of years ago, seeing the need to have data that you can show comparability between different sites. We had a clinical trial with a CD19 CAR with two very disparate sites, one in Cleveland, Ohio, and the other at that time in Moscow, Russia. We couldn't do this study, obviously, today. But we had a generic CAR-19 that we created, and then in both sites, we had the same instrument, the same materials, the same media, the same release reagents, and we found that in both sites, the products were highly comparable, even though the Moscow site at the pediatric center was treating pediatric patients and the Cleveland site at Case Western was treating adult lymphoma. So we showed in that paper that the product is very, very similar, and the vein-to-vein time was 8 days, which meant that every single patient that was enrolled got dosed, got treated, and they all had good outcomes, showing again the benefit of point-of-care manufacturing and making things rapidly and locally. So this is a little bit more color around those clinical trials. You can see on the top left there, that's the team at Case Western. And at that clinical trial on the top right, you can see that we had an overall response rate of 82%, which is great. At the pediatric trial on the top right there, those are patients from two different families enrolled on the pediatric trial. And at the pediatric center in Moscow, a 90% overall response rate. Great. And I'll focus your attention just briefly on the bottom panel. They were very excited. Obviously, they had a press release where we did a bispecific CAR. and targeting two antigens on the leukemia and lymphoma cells. Because what we know is about half of the patients after receiving CAR T-cell therapy relapse. They lose the antigen, right? So when you have two targets, it's much more unlikely for the tumor cells to escape. And in that trial, we had a 92% complete remission rate and only one patient relapse after 2 years. Right? So we were very excited by that. And we're going to continue that theme. You're going to see that the Caring Cross products that we are developing is a triple CAR, a 19-20-22 CAR, to really address the issue of relapse long term, to put it to bed. So when you think about a point of care manufacturing, you think about facilities, right? And the facilities that you can have on the top left up there are either clean rooms that are already existing in hospitals, right? Hospitals have them, they can use them to manufacture CAR T. But there are hospitals that don't have them, right? They don't have access to clean rooms. And so we're working with a company in Florida that makes these mobile clean room units on the left in the middle there. Let me see if I can get this. Yeah, there, right? Germ-free makes these mobile units, which you can actually put them in the parking lot of a hospital. All you need is power. That's all you need, an internet connection, right? Because the amount of fluids that you use is very small. It's just media. And the way that it's organized, you have a reference facility, you can see on the top up here, right? And then you have manufacturing point of care facilities. And this is the model that we're going to be implementing here in Brazil, right? So actually 3 units are now going to be manufactured in the US. One is going to be deployed at Ohio State as the test unit, right? To be able to work out all the final protocols and everything. And 2 units are going to be deployed here in the Rio de Janeiro area. This is what one of the units looks like. I'm not going to spend a lot of time. Suffice to say is that up here you have manufacturing, and down here you have QC. So QC and manufacturing is all very close by, because what you don't want to do is you don't want to manufacture a product and then go to the other side of a hospital to do the test, right? You want to be able to do release testing real time, close, to make sure that you have a very consistent result. So access models and affordability. How are we different than others in the field, right? So if you look up there, this is your typical biotech pharma model, right? They make innovative products that displace current therapies. They have novel IP that protects a high price and market share. They are focused on value-based pricing models to justify the high price. They talk about, the whole package of cost in order that they can charge a high price. And they're motivated to justify the high price to keep full control of the product. And will only develop therapies that are fully controlled by the company and bring a rate of return that satisfies the shareholders. There's nothing wrong with this. This is the way that biotech works. And that's why you see biotech making choices for products that are profitable and not for those, for example, today we've seen with vaccines, that are not profitable. This is the corporate world, and that's fine. Our model's a little bit different. We're a non-profit. We're focused on making just improved therapies, not novel. We're not so worried about IP, but we are focused on getting freedom to operate, right? So that there's no IP that prevents us from making these products. And we can do this based on our knowledgeable team. We know the patent landscape, and there's a lot already in the public domain. Gene therapy has been around for 20 years. A lot of the ideas have already been out there, and so all that is kind of expired. It's already in the public domain. Right now, if you want to get a patent in the gene therapy space, it has to be very specific. It has to be either sequences or something. So if it's sequence specific, there's always a way to find around it to be able to create these products. We're focused on cost reduction, affordability, working with public health systems, for example, Medicare and Medicaid in the US. We're motivated to reduce the cost of the materials and democratize the process. And we're more aligned with hospitals and public health systems to really lower the price and broaden access. Oh, sorry, I went back. And this is what we do, right, in a nutshell. On the left there, we're focused on manufacturing innovation, whether it's lentiviral vector technologies, which my team and I have been doing this for decades, cell manufacturing, the materials and reagents used in those processes, and then assay development. And on the left, you'll see some of our collaborators. Clinical candidates, leukemia, lymphoma, multiple myeloma, HIV, sickle cell, beta thalassemia, they're our top candidates. Some of our collaborators on the right over there. And then the logistical systems, the mobile clean room unit design, the point of care logistical systems of the networking, and then also AI data integration. And there are some of our collaborators there. So we're very focused on developing a robust, very simple process. I'm going to show you the process in a few slides here, but really taking a product, whether it's apheresis or blood, and that's an important discrimination because blood is a lot lower cost than taking apheresis. Selection, transduction. Now, for obtaining vector, we couldn't rely on a third party to make a vector for us that was cheap, right, or affordable, because they would charge us a lot. So we know how to make vectors, so we set up a company called Vector Biomed. I'm going to share a couple of slides on that company with you. Then cell expansion, the candidates themselves, and then the logistics. And this is kind of our model, overall model, that you have a set of reagents, you have a vector, you have specific testing reagents, and that's distributed to centers that have a similar platform worldwide. They have the same instrumentation, and they have the same basic kind of reagents, like the medium, PCR equipment, and so forth. So that's our model, to keep things consistent throughout the network in the world. And this is some slides on our partnership with Fiocruz and Biomagninos. We're very excited by that. And here we are technology transferring everything we know. So we can build local capacity here in Brazil, not only for Brazil, but also for Latin America as well. So we're transferring both the CAR T-cell technology and the lentiviral vector technology. The cost will be about $35,000 per dose, not $375,000 per dose, and it'll be made available to the public health system without significant cost to the patients. The technology transfer is exclusive, not only for Brazil, but for all of Latin America. So we hope to be able to create an innovation hub here. And Fiocruz and Biomagninos will serve as a reference facility for clinical sites in Brazil and in other countries in the region. And we're envisaging not just a couple of products, but a pipeline of products here over the course of the next years. And I just want to acknowledge the vision and the support from the Minister of Health, who saw the importance of bringing these technologies to this country. And we're certainly very excited to be part of this collaboration. And the technology transfer has already started. A couple of weeks ago, some of the team members from Fiocruz and Biomagninos came to visit us, and we're already starting those initial first phases to be able to technology transfer the first part, which is the CAR T-cell manufacturing. And also, we just announced a couple of days ago that we're collaborating with a company called Immunoact in India. This is a company that did the very first CAR T approval in India. And so we're partnering with them to provide our TriCAR so they can bring it to patients in India and for commercialization. So very excited about that collaboration as well. And we're working very hard on a point-of-care network in the USA. The US, compared to many other countries of the world, has a very different healthcare system. You have a national healthcare system with the option of private payers, right? In the US, it's very fragmented. It's mostly private payers, and you have Medicare and Medicaid as kind of a safety net support for those that can't afford it. So the problem is many of those patients on Medicare and Medicaid don't get access, right? Because Medicaid and Medicaid, as you see there, will pay $257,958, I want to be precise here, for the cell product plus the patient care. Now, if the product costs $375,000, it's underwater, right? There's not enough money there to support the product and patient care. We believe that our material cost, as I'll show you, is $20,000. The lab and facility costs $10,000 to $20,000. So in the US, with the higher labor, we'll probably be in the $40,000 to $50,000 range to manufacture the product, leaving a price that you could be charged of about $100,000, leaving another $150,000 for all the clinical costs, which makes it viable in the US. So we are actively recruiting top centers at the moment in the US to develop this model. We're very excited by that. And just a couple of slides here on Vector Biomed. So we couldn't rely on somebody else making vector for us. So we said, we know how to make vectors. We're going to spin our own company. We're going to get some VC money around it, right? But we're also going to have a relationship with this company that it will make products for everybody else, actually about half price because of the efficient technology. But we'll have a special deal with Caring Cross to make these vectors for us at a much lower price so that we can work with others, to provide these vectors at a much lower cost. So I'm not going to go into great details. All that is that we're in the same facility with Vector Biomed in Gaithersburg here. Fantastic team. And I'm just going to show you the titers of the vectors. Normally, you'll see titers of lentiviral vectors, final titers in the multiple 10 to the 8s. We get that in the harvest in the very beginning. So when you get such a high titer in the harvest, when you go to 10 to the 9, which is usually your target dose, you only have to concentrate about a 10-fold. That's really good because when you don't have to concentrate a lot, you then get a lot of aggregation, which means your yields are much higher. So we have a new technology here, and this technology will be transferred here to the Fiocruz team. And what the company does, it really provides speed, quality, and value for these products. And I want to spend one slide here on the economical manufacturing process that our Fiocruz colleagues were able to see just the last couple of weeks ago. So on the left there, you see we can start with whole blood or a leukopak. You have negative selection antibody that we've developed that you added in a single step. You get purified T cells. You put them in a G-Rex, add your vector, activation reagent, incubate for 7 days, put it back onto the Sativa Cepac-C Profa to formulate, and it's ready for infusion. Very inexpensive, and the material cost for this, including the vector, is about $20,000. And you get a very high cell recovery here, basically not only high T cell purity when you compare to positive selection, but also a high level of gamma delta T cells. I'm running out of time here, but I'm just going to flick through the next slides very, very quickly. And basically what we have here is a very high percentage of stem cell memory cells. And we have a therapeutic pipeline of candidates. I won't have time to go into them in great detail, but we have a triple leukemia lymphoma CAR, HIV, sickle cell. These are our top candidates. As I mentioned to you, a triple CAR is better than a single CAR. These are animal experiments that show you why it's better. So these are animals that have received 4 different populations of cells to mimic relapse. If you look at here, the monocars, they get relapse. You have a triple CAR, no relapse, right? So here we have just some candidates, some very nice candidates showing good transduction efficiency, very good killing of the target cells. And this is some of the animal data here that we have here. You can see the CX170 here completely obliterates the tumor into the animals. And not only that, when we took these animals and re-challenged them again with the tumor, completely resistant again. So we're very excited about this product, taking it into the clinic, first in the US, and then we'll bring with some of that clinical data here in Brazil. HIV, I'm not going to go into it. There's not enough time. It's a duo CAR. We're in the clinic now. We're very excited to go to our next level of patient dosing. And for sickle cell and beta thalassemia, it's a very important disease, particularly in this country, especially in the Northeast here. But the price of the product is very, very expensive. And just to show you that this kind of product is curative. There's already a product on the market. It's a lentiglobin for sickle cell. You can see on the left there, the vaso-occlusive crises, that's all in purple. And then after therapy, no vaso-occlusive crises, right? So in beta thalassemia, right, you only need a single dose for sickle cell or beta thalassemia to be able to cure the patient for the disease. But the price is very expensive, $2.8 million. We're going to do it for much less than 1/10 of the cost here in Brazil. This is some of our preclinical data. It looks very good. We're very excited. And just one thing about our team, we're not just a people that have come into this field. We've been in this field. This is all that most of us have been doing for the last 20 or 30 years. So we have a lot of experience. We want to share that experience and make sure that as many patients as possible get access to these transformative therapies. And I won't go into the summary. I just want to thank our partners. This takes a village to do this. Actually takes a city to do this. And so partnerships for us are very, very important. We can't do this alone. Nobody can. And so that's why we're very excited with the collaboration that we have right here. Thank you very much. I appreciate your time.
Thank you, Boro. And now we have Martín Bonamino, who is going to talk to us about the use of those therapies in our public health system.
Okay, thank you, Antonio and Boro and Jim, who's not here. So this is kind of a tough topic to talk about gene therapy-based approach to treat patients in the SUS, because I think we have very few examples, if any, of this kind of therapy, especially for in vivo gene therapy. Most of the patients have been tried or have been treated now by using commercial products with prices such as those listed and mentioned by Boro and Jim. So this is a big challenge, actually. And I think the effort we're doing on several institutions here in Brazil aims to cover this kind of gap and try to offer this kind of therapy for most of the patients at an affordable price. So that disclosure made, and I think if we do this discussion again in 2-3 years from now, probably we'll have different things to say. But I will basically attach to the CAR-T model because I think this might be like a sandbox where some alternative has been tried and are under development, such as this, now under Biomangin's coordination on CAR-T cells and also for AAV-based vectors. But especially for CAR-T cells, we have a much richer environment to discuss the kind of approach we can take here in Brazil, right? So we'll be talking about mostly this right part of the complexity for cell therapy. So those will be genetically modified T cells. And just to remind all of you, we have more than 30,000 patients treated currently globally with CAR-T cell products. So very safe, very effective, very costly also. So this is what we have to tackle here, right? And of course, we are seeing this increase in complexity for these therapies. So Boro just mentioned that we're trying to keep this as simple as point of care and economically available or affordable approach. But the truth is that we are now seeing products that are derived from gene-edited cells, cells derived from iPSC cells, and we'll have like a myriad of different products being used here. So I think there will be room for many, if not all of these alternatives. But at this time, I think we have some examples for SUS based on more canonical CAR-T cells that are the autologous cells that are gene-modified with viral vectors or not, and then use it in autologous approaches for the patients. So I think this is no longer necessary to define the CAR molecule, but this is a chimeric molecule, and I think this is very important to highlight that this is kind of the crystallization of decades of development and knowledge on immunology and biotechnology. So it's a very beautiful example of what we can achieve once we're deciphering some biological mechanisms. And the other important thing here is that we have several flavors for CAR-T cells, so different modules such as the antigen binding domain, the signaling domain, and even the hinge and the structure of CAR-T cells can be changed. So as Boro just mentioned, we can design and redesign CARs and find ways of getting off the IP issue, so we can have freedom to operate if we design and combine different of these domains. So this is important because if we're about to develop new CARs, we have to know how to do it and have freedom to operate to offer this to the Brazilian population, right? So this is a short list of the products that have been approved. The 4 in red are also approved in Brazil. Most of them will have a tech price of about 2 million reais, which is clearly not reasonable for the public system. I think neither for the private system here. So all we have heard today is about how to do this in a more affordable way. So I'll try to condensate some of the concepts and solutions that we're seeing in the country, mostly for CAR-T cells. But I'd just like to mention that for producing any of these advanced therapy products, we need like a change of goods and starting materials it's not available in Brazil yet. So from simple plasmids and DNA fragments to RNA, high quality RNA, and let go viral vectors. So we have some initiatives here. None of them is yet treating patients with virus or viral vectors produced in Brazil. So we think that in 2 years from now, 1 year from now, we'll have a new landscape. But this is the situation now. So we need desperately to have a change of production of goods and starting materials established in Brazil. And I think having big partners such as Fiocruz, Biomangin's, Butantan, and many others can be a good way to make this happen faster, right? So you have different ways of getting access to CAR-T cells. One of them is ordering from big pharma. So I'll make an analogy here with the coffee, and you know this brand, but so you can say your name, they can make the cup of coffee, write your name, and deliver it to you. You can prepare on these automated machines, mostly like we do with coffee. You buy the capsules, you push the button, the machine will do it, all the process by itself. And you can also produce or develop. So here you seed the coffee, crop it. Maybe you can have some help from our Jacu friend here, and then have to collect them, wash them, right, roast, and then you can prepare your final coffee portion, right? If you put it in a perspective of production, you have like users, producers, and developers, such as you do for softwares and many other things. The same is true for CAR T cells. So now we have a very complex landscape in Brazil, many private centers using or buying CAR T cells. Some centers starting to produce them locally with automated machines and or platforms, and some few groups developing things. So you just saw the presentation of a student from LOA's groups. There are many groups in Brazil developing things. None of them, or few of them, have the capacity to take it to the clinics. So closing the whole cycle of conception, development, and first in human testing is very challenging. So very few institutions can do this nowadays in Brazil. So if we can classify those centers, we'll have some producer centers, some of them using some automated platforms, such as the Prodigy platform from Maintaining. So we have the Emocia at Ceará State, we have the INCa Institute in São Paulo, we have some machines that have been already installed and deployed to Butantan, Ribeirão Preto. So those centers will produce or can produce now in these automated machines, but we have a very important bottleneck to getting these viral vectors, certified GMP compliant viral vectors to, and this is one of the goods that or starting material that you have, or one of the main materials that you put in your culture to transduce the cells that get them CAR positive. So some groups are teaming up with teams like Barcelona group that has developed an in-house manufacturer for the vectors. Some of them are chasing Boro to get the vectors here in Brazil, and some of them are getting them from other centers. But this is a very complex situation where sometimes you buy the car, let's put it this way, but you have no fuel. You have some other developer centers such as those that will use closed systems. So we have the example of the INCa Institute, which have developed a manufactured protocol based on an automated system, and some others that use either automated and manual systems, such as the groups from Ribeirão Preto. And then you have some of the local point-of-care initiatives that we have heard now. So in the first moment for CAR T cells in Brazil, the patients were sent abroad. Then we have some clinical trials from industry, and then we had the approvals for these products and patients being treated in Brazil, but products that had been manufactured elsewhere. And now we have some academic clinical trials approved now, 2 of them in Brazil, one from the INCa Institute and the other one from the USP Ribeirão Preto or Hemocentro de Ribeirão Preto, already recruiting and treating patients. So finally we get the local initiatives treating patients in Brazil. And we have many challenges and opportunities to develop these therapies and apply them and offer them to the SUS, our health system. One challenge that we have an extensive network of blood banks and cord blood banks. This is a challenge in a certain way because now we have to decide what to do with these institutions, but on the other hand, we have several groups that have been trained now to do autologous and some of them allogeneic bone marrow transplantation. This might be the ground to apply very safely CAR T cell therapies now. We have a network of small labs that had been established a few years ago that were very important and are still important to try some proof of principle approach to new therapies that includes potentially CAR T cells. We have a huge country, so this is a huge challenge, how to offer this to several different regions of the country. We have the SUS, that's a big buyer and can negotiate prices. And we have also some viral vector supply challenge that hopefully some initiatives such as the one in Ribeirão Preto, the one in Biomanguins will start to taper by offering some of these vectors at least. And we have some opportunities. Many of them are the same on the list of the challenges because we can take this challenge and turn them into opportunities. So we have this capillarity of centers treating patients with cord blood, with transplants, and many other things. We have experimented centers for bone marrow transplantation that might be very important for managing CAR T-cell toxicity and many other things. And of course, we have the capacity to produce some of these goods. So we have these huge, important institutions such as Biomanguins, Butantan, who have shown that they can engage and produce vaccines, biologicals, and now, why not, some of these viral vectors that we need. DNA, high-quality DNA, viral vectors, and so on. So one thing about CAR T cells is that they are just like any high-technology device where you can assemble and embed many technologies in. So you have the possibility of assembling CAR T cells in different ways that gives you some freedom to operate, that gives you some flexibility, and a lot of room to improvements and evo designs. So this is something that we have been trying to do in our groups, and many other groups have been trying to do in Brazil as well. We have now many CARs approved. Most of them will be against the CD19 target, not the triple CAR that targets not only CD19 but also 20 and 22. But we have BCMA and CD19 as solo single targets being targeted by approved products in Brazil. And of course, if we want to innovate here, we have to design and validate new scFVs and new antigen recognition domains. So this is why we set this initiative between INCa, Fiocruz, and also the Brasilia University that we call like the CAR T of Mucuripe. Mucuripe is a harbor in Fortaleza, right? So we have some of these representatives here. Giovanni is here, Marcos Lorenzoni is here, and the UNB team, where they have designed new scFVs, and we are validating them so we can have freedom to operate, to combine them in different ways, right? So we already have some new anti-CD20 scFVs validated here, some anti-CD19 CARs validated here as well, and so they behave just the same as the benchmark FMC 63 CAR. That's one of the approved CARs being used in commercially, or most of the commercially approved big pharma-based CAR. So here you can see that you can innovate, you can copy, you can do like a generic domain, and this is very important because then you have freedom to operate. Of course, they also work very nicely in vivo. And again, assuming that we have to try this, we should make like viral vectors, spend a lot of money, even if we want to try them in the first human trial with very limited number of patients, like 8, 10 patients. So we're very committed to develop new ways here to push this to a first-in-human using like a fraction of the cost. That can be by providing viral vectors at lower cost. That can be by providing solutions that are non-viral vector-based, such as the work Leonardo just showed to you all in the previous session, right? So the production models is also a very important topic here. So this all started in academia, went to very small labs, validating the first CAR products, then it was bought and acquired and incorporated by Big Pharma. And then not only Big Pharma, but everyone's trying to find and to develop consistency. And now some of these automated machines are making this much easier. But on the other hand, they are also making much easier to local centers that treat the patients to try to produce the CAR T cells at the point of care. So we're having like a movement of going back to decentralized production. And Boro just showed an example of how it can be done. Some of these platforms are so I'm not advertising here, so that's why I brought 3 of the main of them, but now we can expect to be flooded by different devices that can do pretty much the same, prepare the cells in different levels of automation, fully automated, semi-automated, and many other options. And then you can see that many groups will try to establish this in their hospitals and start preparing the CAR T cells using the materials. The other thing that's very important, as Boro just mentioned, is the viral vectors by itself or by themselves. So this can be very expensive, but also the viral vector and the CAR structure means that for each one of the CAR domains, you can use different molecules. And again, if you just combine these alternatives, you see that you can find several, maybe dozens of different CAR configurations and have freedom to operate in this design as well. Going back to the viral vectors, this is how much it costs in a facility abroad in the US or Europe. You can take more than 1 year to get a slot to produce your new CAR. So assuming that I have a brilliant idea, it will still take me like $1.2 million, more than 1 year until I get a GMP-compliant viral vector. So this is not a good way to innovate, right? And it's also a full-time job to do this. It's not something that any amateur center can do. So that's where our production units, so I'm listing 2 of the units, but we have Senai Cimatec, Milena is here. We have other units that can play roles in producing this kind of vectors. So this is very important for us. So I'll just end up showing some of the models to approach the local CAR T cell production, right? So this is the industry model. Centralize it, so you collect here, you send it to Netherlands or the US, depending on the company. And then you have to get your own cells, hopefully, not mixing up back. So you can use them in your patients in your center, right? So it's very complicated, as Boro just mentioned. You have a big slice of this cost that's dedicated to logistics and compliance and many other things. There's a local or regional approach on the development in São Paulo. That's the Ribeirão Preto initiative and the São Paulo Capital City initiative, where there's a very nice production center that has been established. Patients have to go there, cells will be prepared, and they can be treated. It's a hub and spoke, but locally implemented, so it's very innovative, powerful initiative, but still patients have to go there. So you cannot imagine a patient from Acre and the patient's family moving to São Paulo for many months. So it's very complicated to get access to this in a large scale. São Paulo is a large state, so it's already a challenge to cover the São Paulo population by itself. And again, we have the initiatives that are not local in terms of this is the Carioca initiative we always play. So this is the car with a view. I bet you will not find a place with a better view to produce CAR T cells than this one. But this is a local initiative, not in the sense that we will cover only the local initiative or the local population, but this is a way of thinking, developing here the initiatives that are based in Rio, but they are developed already thinking about implementing this nationally, right? So let me just mention some of them. The non-viral CAR T cells that have just been shown by Leonardo. So this is the pipeline. This is a very cost-effective way of preparing the cells. We're planning to run a phase 1/2 trial at INCa with this initiative any moment in the next months. The manufacturer will be held at the National Institute of Cardiology, where there's a certified clean room. So this is a joint initiative. Another initiative that it's running now, it's the Children's Hospital of Philadelphia and INCa agreement, where we are establishing now a tech transfer with the very same protocol that has been used to treat the first pediatric patient. That's Emily Whitehead. Most of you probably know this story. And we're teaming up with this team, and they have been changing their manufacturers for an automated system. That's the Prodigy platform with their own lentiviral vector. They're transferring this and we'll be treating about 30 patients with acute lymphoblastic leukemia, pediatric patients. So this is a way of speeding up things, and maybe, since this is a closed system, it could be an adapted way or an adaptation that we could take here and adapt most of these cord blood banks that are distributed across the country and that are not being used that much because, you know, the cord blood used for transplantation is going lower and lower, and so we have to think about how and what to do with these facilities. And last but not least, the Fiocruz Caring Cross / INCa also initiative that Boro just mentioned. And this is, again, a very interesting way because we're just teaming up two federal institutions that are located in the same place to try to validate this product and offer this later on to the SUS. So Boro has just shown the power of having this triple CAR design to avoid escapes from leukemia. There's another initiative that will be run in Biomanguinhos/INI, that's the Infectious Disease Institute here. That's a dual CAR for the GP120 target for HIV infected cells. But the thing here is that there will be this transfer of technology to produce the vectors. So this should make everything much easier. So having the containers is a very important step. This can be replicated in different places. So here we'll have a container at INCa and one container at Biomanguinhos to cover the Leukemia Lymphoma Initiative in INCa and HIV initiative in Biomanguinhos. And this model certainly is a hub spoke, but it can be replicated in a national level. So the idea here is that once we have this running here, several other states in Brazil can receive different containers with the same structure, the same quality control check, and compliance coordinated by Biomanguinhos, and why not to Latin America, as Boro has mentioned here, so it can go beyond Brazil. Right, so this is my last slide. Since I showed some of our initiatives, I'm just listing the people from my lab here. I'll just leave a huge acknowledgement to the institutions and funding agencies that had supported our efforts. And here I mean not only ours, but also others that we are developing with groups in Fiocruz and other institutions. So thank you very much.
Thank you, Martin, for exploring the many possible ways to bring those advanced therapies to our public health system. And now I think we have Marco Krieger online to discuss the presentations, and we won't have time for questions after Krieger's talk. Please, Marco, go ahead.
Sorry. I will try to do in a very fast way. So first of all, I'm really pleased to be able to participate, even in a virtual way, in this important section of the International Symposium of Immunobiological, sponsored by Biomanguinhos. So I think the pioneers' force of these different research that are all collaborating with Fiocruz in some different way in the field of advanced therapy, in addition to our production capability in Biomanguinhos, I think we will allow us to develop alternative strategies to guarantee access for our public health system patients to these new therapies. I think this is the important thing that we are trying to develop, and I would like to highlight the efforts of all teams that are involved in this project in Biomanguinhos and in Fiocruz, the legal team, the technical team that are working the project, as well as all the colleagues that are working this complex project. So I think in this panel we have Antonio Carlos that are working with us in the coordination of this project. We have Martín that is also belong to Fiocruz, but work also in INCa have a very big contribution to our project and also to different initiatives. We have our international partners, Boro and Jim, that are all committed to the same project. The new developing of a strategy that will allow us to work in the local production of these of the vectors and the cells and the technology for the Brazilian public health system, but also bring to Fiocruz Biomanguinhos an important role at the global level. So I think this is the message that we have here in this important section. So last but not the least, I would like to thank all the organizer of the Congress to allow me to participate in this session, even if I am in another meeting here. And that, I think, was really a very good section and a very good opportunity to show for our colleagues the Fiocruz initiatives in this field. So thanks a lot for this opportunity.
So unfortunately, as I mentioned before, we're running late, and I was told that we have no time for questions. But Boro and Martin will take any questions individually after we finish here, okay? Thank you very much.
Obrigada, Antônio Carlos, pela sua condução. Obrigada a
Thank you,
todos.
Antonio Carlos, for your moderation, to all participants. Thank you. Thank you, guys. Again, don't forget the donations. You've seen that they can be made online. I don't know if you got the QR code. Do we have the QR code here? But please have access to the QR code. Please, if you can bring tomorrow your donations, water, non-perishable foodstuffs, cleaning items, and food for your pets, cats and dogs. Again, we'll see you tomorrow at 9 a.m. Again, thank you. Have a good evening and see you tomorrow. Thank you.